What's going on, guys? Reed here. What's going on? I'm gonna send out a few invites. What's going on, guys? Reed's here. Today, I'm going to go over a group of G proteins called GABA. GABA is the main inhibitory G protein. And I'm gonna go ahead and share. So, what's going on, guys? Hope everybody's doing well. What's going on, guys? So let me go ahead and get started. Get my desktop ready here. Let me go through this. Let's see here. Let me turn the sound down some. Great. So let me go ahead and look on here. We're going to go through G proteins. Let's see, how does this work? Nope, I think this is just a message board. Okay, here's going to be here. All right. Okay. And let me see, is this one? I think this one's larger. Yep. Or is this one larger? Okay, I think I like this one better. Okay, so let's see. Let me look at GABA. Explain how the G protein GABA works. Okay, let me see if we get something simple like that. Okay, so let me give you an overview. So first, there are two major receptors which are classified as G proteins, usually have 7 subunits, and they go through the entire phospholipid bilayer. They transcend the entire phospholipid bilayer, and they usually work together as components to make a very complicated structure known as a G protein. And so GABA there are 2 types of main G proteins that are present in probably every tissue, and that's the main excitatory neurotransmitter GABA, glutamate, and GABA is its opposite. It's the main inhibitory neurotransmitter, GABA. And these 2 sort of work together to keep the organism in homeostasis, right? So when people get exhausted or fatigued and things like that, usually they have an excess of glutaminergic activity. So they're usually agitated, restless, irritable, and maybe combative. That's that excessive glutaminergic activity. It sort of means that the ship is sort of off balance some. And we need to activate GABA, meaning exert more inhibitory tone over the entire organism in order to sort of get the organism balanced. And so we're going to look into the GABA receptor. So here, explain how the G protein GABA works. Okay, G protein activated GABA receptors. Okay, they work by binding to GABA, right, which is a neurotransmitter, right, which then activates a G protein. Let me try to keep this on louder because I think the writing may be larger here, so you can see the writing larger. Which then activates a G protein complex that ultimately slows down neuronal activity by inhibiting ion channels that cause excitation, such as voltage-gated calcium channels, activating inhibitory channels like GABA-activated potassium channels. This process reduces the excitability of the postsynaptic neuron, allowing for slower and more relaxed brain processing and acting as a key inhibitory mechanism for the central nervous system. The key inhibitory mechanism. So, and see the play: glutamate, excitation, GABA, inhibition. All right, step-by-step mechanism here. And I think it's important for us to sort of understand the functionality of GABA because when we see a patient who's overly excited or agitated or combative, then we need to know exactly what our targets are. So we're giving this medication and exactly what is it we're targeting. Now remember where the GABA receptors are located. You know, they're located on that mesocortical pathway, right? Mesocortical hyperactivity, positive symptoms of schizophrenia. Of course, we want to target the GABA receptors there to sort of start decreasing the excitability of that mesocortical pathway. Remember the mesolimbic pathway, the reward center, and everything, hyperactivity in that pathway also has GABA receptors, and we can use the GABA receptors to sort of help reduce the excitability in those networks. Also, remember the networks in the basal ganglia, the overexcitation and things like that, and how we can sort of use targeted GABA receptors on those networks. So when we know exactly where the GABA receptors are located and what networks, when we decide on which medication we want to use, we know exactly which pathways we want to hit, right? And so we give the medications, we can titrate the dose upward or downward based upon the clinical effect that we're seeing. And if you know the networks, then you can anticipate what the symptoms are going to be. So here, you look at the step-by-step mechanism. GABA binding. The GABA, the primary inhibitory neurotransmitter, binds to the GABA B receptor on the postsynaptic neuron. So GABA, inhibitory neurotransmitter, binds to GABA B receptor on the postsynaptic neuron. All right. G protein activation. The binding of GABA to the receptor causes a conformational change in the receptor, which then activates a different G protein, a complex associated with the receptor. This type of G protein has a different subunit than the other ones. Okay. Then the subunit dissociation, two pieces of the unit breaks off and they become active and start chewing, making things. Subunit dissociation, the activated G protein complex dissociates into its component subunits, which is a G protein alpha unit and a beta unit. Right? So you get binding of GABA to the GABA B site, the 3D conformational change. It releases the alpha and gamma subunits. They are released from that complex and then they become active. See what happens next. Okay, cellular modulation. The alpha subunit, this subunit can inhibit adenylyl cyclase, reducing the production of cyclic AMP, which is the CMP, that's the precursor to ATP, which is the energy source, right? Biochemistry, right? the production of cyclic CMP, a common second messenger that promotes cellular excitability. So in here, the alpha subunit inhibits adenylate cyclase, and this reduces the production of cyclic AMP, which is the precursor to ATP, which is the energy molecule. So if you're decreasing that, then I think it makes it less excitable. That makes sense. Let me see what the beta subunit. Beta subunit, this subunit is known to directly modulate ion channels. Okay, great. It can inhibit voltage-gated calcium channels, and can also activate G proteins activated inward rectifying potassium channels, whatever that is. They come up with new channels all the time. GERK, G protein activated inwardly rectifying potassium channels. You kidding me? That's a name? Okay. So anyway, so the alpha subunit inhibits adenylate cyclase and reduces cellular activity, and the B channel basically inhibits the ion-gated. That makes sense. So it's going to decrease the sensitivity of the ion-gated channels, therefore decreasing the degree of depolarization, and therefore the cell isn't that excitable. And then it reduces the total amount of ATP in the cell, the energy source in the cell, by reducing its precursor CAMP. The CAMP right here. Okay, makes sense. Okay, so and these are the two. I guess this is it right here. The mechanism is the alpha adenylate cyclase and the beta. This is the mechanism by which the GABA decreases the inhibitory tone. Okay, next, inhibition of neuronal firing. The combined effect of inhibiting calcium influx and promoting calcium efflux from the cell to a lower rate leads to a slower rate of action potential, slowing down communication between neurons. This here is I think is a very simple example of GABA and its effects. Let me look at the medications and the impact on GABA. So next question, let's see here. Let's see, explain GABA and GABA interaction with alcohol. Okay. Alcohol enhances the brain primary calming neurotransmitter. Let's say inhibitory. Calming. I don't think we use that language in neuropsychiatry. GABA, by increasing its release and boosting the activity of the GABA receptor. So alcohol releases GABA, activates the GABA receptor, slows down brain activity, leading to relaxation and sedation. Alcohol targets GABA. I think it's specifically the B site on GABA. Activate the receptors, slows down brain activity, relaxation, sedation. Chronically, the brain adapts by reducing GABA receptor function, causing tolerance, dependence, and increased excitability during withdrawals, making alcohol use potentially addictive. So let's go through this part here. Chronically, the brain adapts by reducing GABA receptor function, meaning the person has less capabilities of less capable of inhibiting all these impulses over time, causing tolerance, dependence, and increased excitability during drug withdrawals. Okay. What is GABA? Okay, we went through this part here, neurotransmitter, and okay, how alcohol interacts with GABA. Okay, increased GABA activity, and alcohol, particularly alcohol, acts on the GABAergic system in the brain. It increases GABA release. Alcohol can cause more GABA to release from the presynaptic neuron. I was wondering where the GABA came from. It coming from the presynaptic neuron. Enhance GABA A receptor function. Alcohol also binds to, is the GABA A receptors? I think it's GABA B, increase their sensitivity activities. Okay, let me see here. Chronic alcohol consumption. When the alcohol consumption is chronic, the brain adapts to the constant elements of GABAergic transmission. All right, to reduce GABA sensitivity, to counteract the overstimulation, the brain can reduce the number of GABA receptors to decrease their sensitivity to GABA. Yeah, decrease GABA receptors. So overall, you have decreased ability to inhibit things, withdrawal symptoms. This adaptation can lead to tolerance, needing more alcohol for the same effect, and a state of hyperexcitability once the alcohol is removed, because you've downloaded a significant number of the GABAergic receptors. So now there's an imbalance between glutamate receptors, which is excitatory, and GABAergic receptors, which are inhibitory. So we need to, at times, give medications and things like that to prop up the activity of GABA. Okay, I think that's it there. Okay, and so we're at 16 minutes now. I didn't want to go beyond 20 minutes, but I think you got my point there on GABA. And let me see if anyone has any questions at all. If you have any questions about GABA and alcohol, just let me know. And let me go through some of the questions. Okay, Wade, how you doing, Wade? Hey, buddy, how's it going? All right. Dr. Liv, how's it going? Marianne, hey guys. Nayib, hey guys. Okay, iHash, thank you so much, buddy. What disorders arise when GABA or G protein signal goes wrong? Everything. Everything. The loss of the inhibitory tone. Yeah, so everything. Yeah, that's a tough question, but everything. Let me see here. Next. How does GABA's effects on G proteins help reduce anxiety and overactivity in the brain? That GABA receptor and the GABA molecule that's released presynaptically results in inhibition, meaning decreased firing of the postsynaptic cell. And so that's its main impact. Decreasing the activity of the postsynaptic cell should reduce anxiety, it should reduce restlessness, it should reduce all the things that are overactive in the brain. Next, okay, thank you, Moeen, appreciate your question. Hey, Umber, how does G protein GABA signaling affect sleep regulation? Overexcitation is basically the networks being overactive, and they really will have to settle down in order for the reticular activation system to sort of start the process of introducing sleep, the structure and the process for sleep. So I think the overactivity basically interferes with that kindling process in the reticular activation system. But I'm not an expert at the reticular activation system. I don't get a chance to deal with it much. I'm on the clinical side where I give them medications, but I think the overstimulation is the reticular activation system doesn't work properly. And thank you so much for your questions. Okay, so we're at 19 minutes and 2 seconds. I wanted to keep this at least in the 20 minutes or not. So if you don't have any other questions, I appreciate it. Do. So it's a pleasure talking to you guys. And if you have any other questions, thank you for attending my lecture. And you learn G proteins and you take care. Take care, y'all. Dr. Reed. Ciao.